Alrighty, welcome back. So today what we're going to be doing is some MCAT style amino acid review questions. So in the previous two videos that were posted in the Biochem MCAT prep section, we were able to talk about amino acid structure, some functional components, peptide bonds, disulfide bridges, so on. And so today what we're going to be doing is some MCAT style amino acid review questions and seeing if we can apply the knowledge from the previous two videos onto real MCAT style questions. So real quick, before we move into the actual questions, one thing I would recommend for the MCAT in specific is paying close attention to the passages. The test can be long, you're going to be reading a lot, and so it's really easy to just skim over some passages and, just skim through it, try and just read the question and just try and answer it without reading the supplementary material. But I do think that the MCAT is one of those tests where reading the passages is pretty important because there are a lot of hints that you're going to find in the passages that are going to be very useful to answering the questions. And so with that in mind, let's move on to question one. So what I'm going to be doing for these questions is I'll give around 10 seconds if you want to pause the video, try and answer the question yourself, and then I will go ahead and work through a solution with you guys. So question one reads, researchers purified a protein and found that treatment with a reducing agent caused the protein to lose its tertiary structure. So one thing that sticks out right off the bat is that they used a reducing agent and we're losing tertiary structure, which means some sort of bonds are being broken. Analysis revealed that the protein contained multiple cysteine residues. Okay. Which of the following best explains the effect of the reducing agent? And so, what this question is really testing, and what jumps out to me right off the bat, is that it has to do with cysteine residues, it has to do with reduction, and it has to do with protein losing its structure. So that means some sort of bond is being broken. So we're thinking about cysteines, we're thinking about reduction, and we're thinking about losing bonds. And that's what we've really taken from this paragraph. And so immediately, without even looking at the answer choices, we're able to apply our knowledge from the previous videos because we know that reduction occurs when a disulfide bond, so one cysteine with an I, goes back into two cysteines. And so now, before even looking at the answer choices, because we know that reduction causes cysteine, which is a disulfide bond, to go back into two cysteines, which is no bond, we're already better prepared to answer the question. And this is without even looking at answer choices. But now let's go ahead and take a look. So one by one, answer choice A reads, reduction converted cysteine residues into cysteine residues, disrupting covalent crosslinks. All right, well, that's pretty much exactly what we said. So I'm just going to circle this for now and say that that seems like a pretty reasonable answer. Option choice B, reduction protonated cysteine side chains, eliminating hydrogen bonds. And so we know that the cis side chain looks something like this. So hydrogen bonds can occur, with this sulfur and this hydrogen here, but this isn't the main functionality of this R group. And actually the hydrogen bonds that can occur with this style group are very weak. And so they're not as significant for protein tertiary structure or even quaternary structure as are the disulfide bonds. So while the answer choice is a reduction protonated cysteine side chains, eliminating hydrogen bonding, it's really this part that doesn't make sense. Whether the hydrogen bonding is there or not, that's really not going to affect the tertiary structure of the protein. And so I'm going to say that this answer is not going to be the correct one. Moving on, a reduction hydrolyzed peptide bonds between adjacent amino acids. So this isn't the case. Reductions don't hydrolyze peptide bonds. Remember, hydrolysis of a peptide bond isn't redox. So while, hydrolyzing the peptide bond, which means to separate a peptide into its component amino acids, probably would have a significant effect on tertiary structure, this reaction is not a reduction. And so that's why that answer choice is false. And then moving on, a reduction removed the amino groups necessary for protein folding. And so again, the amino groups aren't what's necessary for protein folding. They do play a role, in the formation of peptide bonds, but they're not what's going to be necessary for that tertiary and quaternary structure. They're not as important as hydrogen bonding, or in this case, disulfide linkages. And so this answer choice is incorrect because this is talking more about primary structure instead of tertiary structure, which is what the question is really focusing on. And so the answer to question one is A. A reduction converted cysteine residues into cysteine residues, disrupting covalent crosslinks. And this fits with everything that we were looking for. It involves cysteine. It shows how a reduction breaks a disulfide bond, and then it shows how that disrupts covalent crosslinks. Okay, moving on. Question two. And so now let's move on to question two. So at physiological pH, alanine predominantly exists as, and then four choices. So immediately right off the bat, we're talking about physiological pH, which is around 7. And so really what this question is asking is if we know about the Zwiller-ionic state of amino acids. And so quickly, we can just draw what we expect alanine to look like at this pH, which is with a protonated amino group and a deprotonated carboxyl group. And so this is just one of those things where you need to have a relative idea that the pK of the amino group is roughly equal to 9, and the pK of the carboxyl group is roughly equal to 2. And so, using this information, we see that the pK of the amino group 9 is higher than a pH of 7, meaning this group should be protonated. And the pK of the carboxyl group is 2, which is lower than physiological pH, lower than 7, meaning this group should be deprotonated. And so now when we're looking through our answer choices, we're really just looking for, one answer in which the R group is correct, first of all, so we know it's alanine. The amino group is protonated, and the carboxyl group is deprotonated. And so now let's go through the answers. So A, the amino group isn't protonated, so that one's wrong. C, the amino group isn't protonated, so that one's wrong. B, the carboxyl group is still protonated, so that one's wrong. And that leaves us with one choice, D, which is the correct answer. And so, aside from, or just by reading this question, the one thing that, is really being tested here is whether you know how amino acids act as Zwiller ions, and whether you know, like, relatively the pKs of the amino group and carboxyl group, which are two groups that you should have, not memorized, but just have a rough idea that they have a pK around 9 for the amino group and have a pK around 2 for the carboxyl group. All right, moving on to question 3. It reads, a researcher synthesizes a tripeptide from 3 amino acids. How many water molecules are produced during synthesis? Okay, so what we're doing here is some researcher is synthesizing a tripeptide. That means they're putting 3 amino acids and combining them together from these 3 amino acids. How many water molecules are produced? And so we know from the previous video that if you take 2 amino acids and combine them, this produces 1 H2O. And so basically, per every one peptide bond made, 1 H2O is made as well. And this is because this is a condensation reaction. And so if we're taking 3 amino acids and we're turning them into a tripeptide, meaning we're putting peptide bonds between them, we're now making 2 peptide bonds, which means we should be making 2 waters. And therefore, the answer is going to be B. All right, moving on to question 4. It reads, a mutation replaces a leucine residue located in the hydrophobic core of a protein with aspartate. So we're taking leucine and replacing it with aspartate in a hydrophobic core. Which effect is most likely? All right, so we have a relative idea, or we know roughly, that leucine has the R group of CH2, C, CH3, CH3. And we're replacing it with an aspartate, which has the R group CH2C, O, and then a minus because it is acidic. And so just looking at these two R groups, we're going from something completely, hydrophobic, it's purely hydrocarbons, and completely nonpolar. And we're replacing it with something, arguably of similar size, but now it's polar. It's polar, it's acidic, and it even has a negative charge, right? And so what would you expect to happen if this, aspartate replaced this leucine? Well, just looking at the difference between the R groups, it's pretty safe to say that because this is occurring in a hydrophobic environment, you're going to have some destabilization. Negative charges or charged molecules in general do not want to be in hydrophobic environments. They want to be exposed to solution and therefore they can be stabilized by solution. And so if we're forcing a negative charge into a hydrophobic core, it's safe to say that destabilization is going to occur. And so let's read through the answer choices with this in mind. So we're increasing hydrophobic interactions. Well, that's not going to be right because we're removing something hydrophobic and throwing in something hydrophilic. So that's not going to happen. Answer choice B, we're increasing the stability of the protein core. Again, we're looking for destabilization. So that's not going to happen. C, destabilization due to the introduction of a charge residue. That seems correct. And then D, formation of additional disulfide bonds. Well, that has nothing to do with the problem because neither leucine nor aspartate can make disulfide bonds. And so therefore, our answer is going to be C. Moving on to question 5, it reads, a polypeptide contains 100 amino acids. Complete hydrolysis of the polypeptide will require the addition of approximately how many water molecules? And so unlike, 2 questions ago when we were talking about how you can take individual amino acids and put them together, it takes as many waters, or you will produce as many waters as you make um, peptide bonds, right? But now the scenario is the opposite. Let's say we have 3 amino acids in a tripeptide. In order to break each individual peptide bond, you are going to need water. And so per peptide bond, you're going to need 1 water to break it, because that is how the hydrolysis reaction works. And so if you have 100 amino acids all string together like a bead in a chain, that means you have 99 peptide bonds, meaning you need 99 H2Os to destroy them. And so that is going to be our answer, 99. So one quick tip I have for questions like this is if you're given, a polypeptide with X amount of amino acids, the number of waters you're going to need either to create that polypeptide or, like in this case, to destroy it is always just one less than the number of amino acids. And so that's, a quick tip. It can really save you a lot of that time on the MCAT, which always will be helpful. All right, moving on to question 6. A researcher combines 4 molecules of cysteine under conditions that allow complete oxidation and peptide bond formation. All right, so let's stop there. We're combining 4 molecules of cysteine and we're allowing for oxidation and peptide bond formation. So let's just draw out what that might look like. At least in terms of peptide bonds, this is something like what we might see. Moving on, the final product consists of a single linear tripeptide, which is what we've drawn out, containing 1 intramolecular disulfide bond. All right, so let's take a look at that. Let's say we have a linear tripeptide, or sorry, tetrapeptide of cysteines. We're now introducing 1 disulfide bond, so it could look something like this, with this being the disulfide bond. All right, relative to the 4 free cysteine molecules, formation of the final product results in a net production of. Okay, so now we're talking about the net production of what? So we know that a peptide bond is being formed up here, and we know that oxidation is occurring, which means that a disulfide bond is being formed because oxidation and disulfide bond formation go hand in hand with each other. So let's tackle this problem one aspect at a time. So we have peptide bond formation and we have disulfide bond formation. We know that for every peptide bond produced, we're going to be making a water. And so if we're making 3 peptide bonds, that means we expect to be making 3 waters. Now, for a disulfide bond, per disulfide bond, you're expecting to produce 2 H protons, or 2 hydrogen protons, and 2 electrons, because this is an oxidation. So you're losing electrons in the formation of this bond. And so since we're only making one bond and one disulfide bond and 3 peptide bonds, these are the products we're expecting to see. So now let's look through the answer choices and see, if any of these answers align with what we expect. So answer A says we're making 2 hydrogen or 2 waters. Well, this doesn't align. This would be the case if we were making only 2 peptide bonds, but we know that we're making a tetrapeptide, so we need 3 peptide bonds. So that's not the case. And then for similar reasons, D isn't the case because we're not making 4 waters, we're expecting to make 3. And so now looking at answer choices B and C, they say 3 waters, 3 waters. Okay, that's the same. 2 hydrogens, 2 hydrogens, that's the same. The only difference is that C says we're making 2 electrons and B doesn't have that. And so, like I said, when you're making a disulfide bond, this is an oxidation reaction, which is hinted at right here. And so something is being oxidized. That means electrons are being lost. So electrons should end up in the product side of the reaction. And that's the reason why C is the correct answer. We need to see those electrons because this is the result of an oxidation reaction. All right, moving on to question 7, it reads, a globular protein contains the following buried residues within its hydrophobic core: valine, leucine, phenylalanine, isoleucine. And so right off the bat, we can stop right there. We know that, these are hydrophobic. Phenylalanine, we know this is aromatic, and we know this is hydrophobic as well. And so, it's always good to, with these type of questions, especially MCAT questions, just to stop as you're reading something, you just, draw something down, get the picture straight, and it'll really help you as you're moving through the questions. So let's move on. Which mutation would be expected to produce the greatest destabilization of the folded protein? So if we're looking for a destabilization, we don't want to replace something hydrophobic with something else hydrophobic, something aromatic with something else aromatic. The greatest destabilization will be produced when you take something, hydrophobic, for example, and replace it with something hydrophilic. So if you want to make destabilization, you really need to take something and replace it with its opposite. That's going to cause the greatest destabilization in a protein. And so now let's look at the answer choices, keeping that in mind. So answer A proposes that we replace valine with leucine. Well, we know that both of these are hydrophobic. And so, while they may vary slightly, it's not really going to cause a huge destabilization of the folded protein. Answer choice B says replace a phenylalanine with a tryptophan. Well, this is the same thing. We're replacing something aromatic with something else aromatic. They might differ in size, but you're probably not going to see a huge destabilization. Answer choice C, isoleucine to alanine. Again, two hydrophobic things. Replacing one with the other, you're probably not going to see a huge difference. But now, a leucine to aspartate, you're probably going to see a pretty big difference if you replace a leucine, which is hydrophobic, with an aspartate, which is negatively charged, hydrophilic and polar. And so that's why the answer to question 7 is D. All right, so let's move on to question 8. So question 8 reads, a purified protein is completely hydrolyzed into its constituent amino acids. And so just to set a picture or draw a picture for ourselves, we have some sort of polypeptide and it's being turned into its constituent amino acids. So you're going from a polypeptide to multiple single amino acids. Analysis reveals that one of the amino acids does not rotate plane polarized light. Okay, does not rotate plane polarized light. So immediately when you think of rotating plane polarized light, you don't need to know anything about the physics behind it, anything about how this happens, why it happens, whatever. The only thing you need to know about rotating plane polarized light is this is something that only chiral compounds can do. And so if it doesn't rotate plane polarized light, this compound, whatever it is achiral. So which conclusion is most justified? Well, let's take a look. The amino acid existed as a zwitterion prior to hydrolysis. So this doesn't really, it doesn't make sense. It doesn't really have anything to do with the question. So not rotating plane polarized light means that an amino acid is achiral. So whether it's a zwitterion or not, or the charges in general don't really matter. So it's not answer A. B, the amino acid contained a sulfur atom. Again, having a sulfur atom has nothing to do with the chirality of an amino acid. So we know it's not answer choice B. C, the amino acid lacked a stereogenic alpha carbon. Okay, that sounds reasonable. Let's keep that in mind. And then D, the amino acid was originally located at the N-terminus. Again, location at the N-terminus, C-terminus really has nothing to do with whether something can rotate plane polarized light, whether it's or not it's chiral. And so the answer choice that we're left with is C, the amino acid lacked a stereogenic alpha carbon. And just thinking about our amino acids, we know that sounds pretty reasonable because we know there's one amino acid that is achiral, which happens to be glycine. And so just to quickly draw it out, we can see why glycine had this property of not being able to rotate plane polarized light, and it's because it's achiral. And that's because in order to be chiral, remember you need to have 4 unique groups around the alpha carbon. But this glycine only has 3 unique groups. It has an amino group, a carboxyl group, but then it has 2 hydrogens, which are obviously the same as each other, and therefore this makes glycine achiral and not able to rotate plane polarized light. There we go. All right, so let's move on to question 9. A tripeptide contains exactly one residue of each of glycine, cysteine, and lysine. So right off the bat, just to draw a picture, we know that we have a glycine, a cysteine, and a lysine, all drawn or all connected as a tripeptide. Following complete hydrolysis, the products are separated by ion exchange chromatography at physiological pH. Which amino acid would be expected to migrate most strongly toward a negatively charged resin? All right, so like the question said, all these amino acids are separated by hydrolysis. So now let's take a look at their R groups quickly. Glycine has a hydrogen, cysteine has a CH2 and a sulfur, and lysine has 4 CH2 connected to 1 amino group. So this question is talking about ion exchange chromatography, which is a technique where you separate a bunch of different compounds by charge. And so if we're asking which amino acid would be expected to migrate most strongly towards a negatively charged resin, we need to think about the R groups, which we drew, and we need to think about charge, which we haven't looked at yet. So glycine, it doesn't have a pKa. This is something, we talked about in the first video in this series. So no pKa, meaning it just exists in its zwitterionic state with a net charge of 0. Cysteine has a pKa, and that pKa is around 8. But this experiment is happening around physiological pH, which occurs at a pH of 7, meaning that the thiol group is still going to be protonated, since the pKa of the group 8 is higher than physiological pH 7. And so again, it's also going to be in its zwitterionic state with a net charge of 0. And even if for some reason the pH was different and this group were to be deprotonated, it would give the thiol group a negative charge. And so it would repel from the negatively charged resin. And so this would be like an even worse case for cysteine. And now let's look at lysine. It has a pKa around 10.5. 10.5 is much higher than 7, meaning it should be protonated, and so it's going to have a +1 charge. And so now looking at all three of your answers, or all three of our answers, we see that we're comparing an amino acid with a net charge of 0, another one with a net charge of 0, and then an amino acid with a net charge of 1. And if we have a net charge of 1, that is going to attract to the negative resin strongest. And so we know it's not glycine, we know it's not cysteine, we know they all won't migrate equally because 2 have a net charge of 0 and lysine has a net charge of 1. And so we know that our answer for question 9 is lysine. And finally, question 10 reads, a researcher synthesizes a tetrapeptide consisting of glycine, alanine, cysteine, and valine. The peptide is subsequently oxidized to completion under conditions that do not affect peptide bonds, and then completely hydrolyzed. So, basically what we're doing here, or what the researcher is doing here is they're starting with a tetrapeptide consisting of glycine, alanine, cysteine, and valine. They're doing some sort of oxidation, some sort of oxidation to the tetrapeptide, and then they're completely hydrolyzing it into its components of glycine, alanine, cysteine, and valine. So now we just have those 4 amino acids floating around. Which of the following statements must be true regarding the final mixture? So let's go through. A, exactly one amino acid in the mixture is optically inactive. So we know this is the case. We know that glycine is achiral. And so answer choice A is a possibility. That's a true statement. B, exactly one amino acid in the mixture contains a sulfur-sulfur bond. So, this is one way that questions on the MCAT might try to trick you. They say that oxidation is happening. So you might immediately think, okay, oxidation is happening. That means you're forming a disulfide bond. But you only have one cysteine in your tetrapeptide. And you know that in order to form a disulfide bond, you need at least 2 cysteines. So an answer choice that says exactly one amino acid in the mixture contains a sulfur-sulfur bond doesn't make sense because we know that we need 2 cysteines at minimum to create this bond, and we only have 1. So this answer can't be correct. C, the mixture contains 4 amino acids and 3 molecules of water. So answer choice C, is really testing whether or not you know how hydrolysis works. And so an answer choice that says 4 amino acids is correct. We know that there are 4 amino acids. It tells us that. Hydrolysis is occurring. We're taking a tetrapeptide, breaking it up into its constituent amino acids. But then 3 molecules of water doesn't really make sense because we know that in order to break a peptide bond, so starting with a peptide and then going to its constituent amino acids, we need to be adding water. And so water isn't going to be on the products, it's going to be on the reactants. And so if this said required 3 molecules of water, that may be correct. But since it says it contains in a product 3 molecules of water, that is not correct. And then finally, the mixture contains 2 molecules of cysteine. Well, for the same reason that B is incorrect, D is incorrect. We're specifically told it's 1 molecule of cysteine. And that's why question A, or answer A, exactly one amino acid in the mixture is optically inactive, is correct. All right, and that's it for all the questions I had prepared today. If there are any questions, please feel free to leave them in the comments, and good luck on your studying.